Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Tanja Vogel from the University Clinics Freiburg on this show. Please let me briefly introduce you to our audience. You got your PhD from the Medical School Hanover in Germany in 1997. You then moved on to do a postdoc at the EMRC Human Genetics Unit, Edinburgh, and at the MPI for Biophysical Chemistry in Göttingen, and started your own group at the University of Göttingen in 2005. You then became professor in 2010 and joined the University of Freiburg in 2011, where you are still today. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Okay, that is a very nice startup. And I'm happy to share that basically due to my school training, I was very broadly interested in basically everything. So I had biology as sort of one of these major subjects, but also history. And initially I was planning to become a teacher for history and something else. But at the time I was afraid that I would end up as a taxi driver. And so I thought, okay, maybe I take a subject that is more upfront, and thought that molecular biology would be a nice way to go for. And then I decided to study biochemistry and plain biochemistry. At the time, there were only like 4 universities offering courses, and I was lucky to get admission to the University of Hanover. And there I studied then biochemistry, and retrospectively speaking, I'm really happy that I took this chance decision and that I was then diving into molecular biology, basically, and biochemistry, which is my subject of training.
What about the history of science? Would that have been something?
That is a good question retrospectively as well, but it's hypothetical. So history of science this is the past, right? And now I think I'm more eager to learn about the unsolved problems that are lying there before us. And this is really driving me every day, that I want to unpack the boxes, sometimes harboring the Pandora.
Yeah, maybe we can look at some of the boxes that you already packed and unpacked. Your science centers around how epigenetic modifications instruct stem cells during central nervous system development, FOXG1, and the etiology of FOXG1 syndrome and signaling pathways and their connection to chromatin, obviously. So I want to start in 2010. There you highlighted the contribution of histone methylation in the neuronal specification of upper layer neurons in an AF9 mutant mouse model. Can you talk about this story? This must have been one of the first paper out of your own lab, right?
Yeah, this is basically, that is still part of my postdoctoral training, I would say, because I started the project when I joined the group of Peter Gruss in the Max Planck Institute of Biophysical Chemistry in Göttingen. And there, basically, we were interested to identify factors that showed sort of a regional expression in the forebrain, because this is a highly organized and arealized structure, and we were and are still interested how this develops. And then I came across this transcription factor AF9 or MLLT3. It is translocation hotspot and fuses with MLL, which is histone H3K79 methyltransferase. And at the time, basically, it was has a strong link to leukemia. And at the time, it was not clear how these modifiers to which AF9 or MLLT3 belongs, which under mutation mainly show homeotic transformations what these molecules would do in the brain. And, homeotic transformation during development is also mediated by polycomp protein complex. And this basically attracted my interest in exploring how basically Hox regulators that are not expressed in the brain, what they are doing there. And that basically drove me towards exploring MLLT3, AF9. And for this, I was happy to characterize this mouse model and for the first time show that the histone H3K79 methylation basically has an impact of specifying a specific subpopulation of neurons, which are these upper layer neurons. And that was also my first attempt to do chromatin immunoprecipitation teaming up with the lab of Stephen Johnson at the time in Göttingen. And that was, my first real contact, I would say, with the chromatin by precipitation and some qPCRs.
So talks when it comes to chromatin are mostly about modifications of the histone tail. So lysine 79, is this still considered part of the tail? Or I mean, it's always where does the tail end? Is K79 already part of the core? Is there something special about K79 that makes it maybe difficult to chip?
Yes, multiple questions. So first of all, I would consider that K79 is really in the nucleus of the core and not in the tail. And I think that is also shared by all colleagues working on K79 methylation. It is very well chippable nowadays, so there's not a problem. What poses more a problem, at least for my lab, is to basically show the enzyme DOT1L itself and where it is present, right? So we are mainly working in the mouse, and we think that antibodies recognizing DOT1L in the mouse are not really convincing in all circumstances. But the mark itself does not make huge problems. So we have, and colleagues have generated multiple data sets, right, showing really nice ChIP-Seq tracks, and also doing ChIP and qPCR usually gives really faithful results with the respective controls. So I would consider that this is not a problem. The meaning of K79 methylation, that is, I think, a difficult part, right? So literature is full of reports claiming it's an activating mark, and it is, of course, associated also with active transcription. We usually pick and also report on some contrasting results, which show, for example, that K79 dimethylation specifically is also found at loci that are not active, but are more silenced. Or nowadays, I would say that they are sort of poised, because we see a nice crosstalk also to K27 trimethylation. And this is also not a single observation by my lab, but also other colleagues report on this correlation between the two marks, and that might be an interesting way, basically, to follow up research also on K79 methylation, right? So I don't know basically the meaning, right? I mean, you find the different methylations, mono, di, and trimethylation at different sites. As said, we are mainly focusing on the dimethylation, and I don't know what it is, what the meaning of this methylation really is now, because I think that we sometimes have like conflicting results between genetic knockdown or removal of the DOT1L protein, and then its enzymatic inhibition. So that does not necessarily follow a straight interpretation and the black and white pattern, but it is sort of difficult to untangle. There is no reader for this mark as of yet, only few reports claiming there would be. And of course, there's also no way to actively get rid of the mark as we think as of yet. So it is then the only option, right, to dilute this modification through cell cycle regulation, or to going through multiple rounds of cell division, which then does not play any role in the nervous system if we look at neurons, because neurons are terminally differentiated cells, which do not divide, right? And so that would basically mean once you have K79 methylation at one site, it should stick there, right?
Yeah, so you touched already on many points of my next questions. So one of those would be DOT1L, right? It's an interaction partner of the AF9 protein, and it's, I think you mentioned it just now, the reader, no, not the reader because there is none, the writer of this modification. You started to investigate DOT1L. So what did you find about the function of DOT1L in stem cells? I think you looked at it in neural stem cells, and also in the process of neuronal development.
Mm-hmm. So the function of DOT1L as we looking at all data that we have acquired from different neuroanatomical locations, including the forebrain and the dorsal and the ventral telencephalon, for example, and the hippocampus. We looked in the cerebellum and also in the spinal cord. I have the feeling that DOT1L is really a gatekeeper or a state preserver of a given state, either being a stem cell. So you have to override the information that DOT1L is basically referring to transfer to the next stage, right? For example, you have a stem cell, and the stem cell wants to become a neuron at some point. So you have to actively override the information that DOT1L is given to activate programs that allow a cell to express genes that are necessary for neuronal differentiation. And this basically is an observation that really holds true for all of the systems that we have looked at. But also, if you look at the next stage, if a stem cell has acquired a neuronal fate already, DOT1L is regulating transcriptional programs that are implicated in pathfinding and to really place then the cell into the network that it belongs to, right? That is a peculiarity, I think, of the nervous system development, that after a stem cell has left the active stem cell proliferating state and becomes differentiated, that it also has to migrate to the site where it then finds its last location. For example, in the cortex, that means that you migrate as a young neuron from a part where you are near the ventricle towards the outer surface, right? And that can be quite a substantial way that you have to overcome. Or if you are a specialized neuron and you're born in the ventral telencephalon, you migrate tangentially even longer to find your place within the cortex and within cortical networks. And here we have found that DOT1L also is implicated in finding the terminal place where a young neuron basically then integrates into the system. And that is of relevance, right? That because you could imagine that you have diseases where you have a disbalance between or within a neuronal network where you have overexcitation or underexcitation, right? And this is something that you don't want to happen, because it can manifest in diseases like, for example, epilepsy or schizophrenia or I don't know, or there's a whole bunch of more neurodevelopmental diseases that impact the balances of activity within a neuronal network.
Which would be a very important role, right? I mean, every protein or every factor that is involved in neuronal development is very important and probably very hard to study, because if you get rid of it, it will be not be easy to study, probably.
Yeah, if you are too harsh in your condition and your factor is too important, then of course the organism is not developing properly, and usually you have strong phenotypes which lead to, for example, perinatal lethality. This is also something that most of our mutants suffer from. There's a good question: why do these organisms die? Which is a question that we cannot answer, right? Because they have like smaller brains, but this is not necessarily a reason for not being able to live at all. DOT1L is very important for the entire development. This is the reason why the proper and the full knockout basically does not reach later developmental stages than in the mouse, like 7 days, right? Which is very early before you really have an important organ development being initiated.
So DOT1L mediates all 3 degrees of methylation of H3K79, what we already talked about, and its enzymatic activity is critical to modulate cellular differentiation and reprogramming. We already talked about that. And you also investigated the accessibility of the SOX2 enhancer in this context. What did you learn about the function of this enhancer or about the accessibility?
Yeah, this was also really interesting to see that H3K79 dimethylation is not restricted to gene bodies, where basically it is mainly found, but that within neural precursor cells, where we were working on, but also in cancer cells, we saw that K79 dimethylation is also impacting enhancers. And through attack sequencing, which we correlated with these ChIP sequencing data, we saw that we have the mark specifically at enhancers, and they're impacting upon being present or being removed the accessibility towards SOX2 enhancers. Funny enough, these SOX2 enhancers or all accessible sites upon DOT1L enzymatic inhibition do not necessarily also have the mark. So the enhancer can change its accessibility upon DOT1L inhibition without being marked with K79 dimethylation, which is also another miracle that we somehow have to solve, right? And to understand how that can be.
So maybe DOT1L has another function or another target?
Yeah, I think that DOT1L has its main function really through its interaction partners and acting in complexes. And that maybe the K79 methylation itself is sort of a flagging of telling the system that DOT1L was there. But I'm not sure whether the mark itself has a real driving force itself, right? Because that is, it might be really a bystander and not a cause for the effects that we are observing.
Do you know if there is any connection to H3K36? Because it's also found in gene bodies and is an active mark.
Yeah, there are reports, and we also have some unpublished observations that DOT1L might co-influence splicing, which means that it's also co-migrating with the RNA pull2 machinery, right? We know that DOT1L or the complex that contains DOT1L is also interacting with the CTD of RNA pol 2, so they travel along. And of course, also this H3K79 trimethylation has similar pattern, right? So and for this, I think that at least for some genes where we have this striking methylation within the gene bodies, there is a strong correlation to K36 trimethyl. However, we never saw massive changes in K36 trimethyl upon enzymatic interference with DOT1L itself. So I think that they are really correlating, but not acting causatively together, if one may say so.
Yeah, not. I think I heard this like in one of the last episodes. Not just because they are at the same place doesn't mean they are acting together, right? There must, yeah.
Yeah, absolutely. This is also what we observe now with the K27 trimethyl, right? Where we have indication that EZH2 is affected in some systems by changing its expression, but sometimes it's the recruitment. So there are ways to crosstalk, but I think they are versatile. There is a plethora of possibilities how that can be exerted, and it will be really interesting to learn with novel experimentation, whether it's direct or indirect, right? Whether it's just making space for the other one and not occupying sufficient parts or places of the chromatin, so that other factors cannot access, for example. So this would be a scenario that I could envision that explains many of these crosstalks, which might be indirect and correlated.
So we are recording this in October 2023, and over the course of the summer, like in August and September, 3 papers regarding DOT1, I'm just looking at your PubMed record here, were published. And I think the H3K27 trimethylation information you just shared was part of one of those. So is there something you can share about those 3 studies that you want to share?
For our latest results, this they are basically completely unrelated.
Nice.
The bracket, or that is only DOT1L. So first we have published a story of where we looked at the enzymatic activity of DOT1L during cortical development of the glutamatergic, that means excitatory neuronal lineage, where we used extensive single-cell characterizations. Initially, I was interested of how much of the DOT1L information is basically transmitted to a next cell generation. However, we faced some problems in resolving on single-cell level the ChIP results, ChIP sequencing, and with the advent now of single-cell cut and tag, I think that we can go back to this initial question. But in that story, we were able to basically show that the inhibition of DOT1L leads to an activation of metabolic programs, and that on one side is switching to a state of glycolysis that is a hallmark of differentiated neurons. And on the other hand, we saw that DOT1L regulates asparagine synthetase, which, as the name says, synthesizes asparagine, which is also an important metabolic enzyme for brain development, because mutations of that result in smaller brains called microcephaly. And there we could show that this inhibition of DOT1L leads to the reduction of K79 dimethylation, but also to K27 trimethylation, which then basically allows that this gene gets activated prematurely, probably in stem cells, which is followed by differentiation. This is also what we have shown then by overexpressing the asparagine synthetase in the stem cells, which is sufficient to drive them out of their stem cell state and to initiate differentiation. And another paper, we basically looked at interneurons, and they have a different origin and a different developmental logic, but they also depend on expression of DOT1L. There, in that study, we also observed that stem cells exit prematurely the cell cycle. They differentiate too early. And by this, they are exhausting the pool of progenitors so that the last neurons that are basically born, there are not sufficient stem cells for them to generate them. And this is why numbers drop. And this is what we have seen when we do an interneuron precursor specific knockout of DOT1L, that we see that the last interneuron population is not made in sufficient numbers. And in the third study, that basically describes the complex, or one of the complexes that DOT1L is part of. And there we looked at NPM1, nucleophosmin 1, which is also an important a molecule for cell biology. It has many functions, and the function that we were looking on was the organization of the nucleolus. And there we basically saw that the knockdown of NPM1 comes together with increased K79 dimethylation levels, which was also observed by other colleagues. And we could show that DOT1L and NPM1 are engaged in a feedback loop of regulating each other's expression, probably to balance availability of the respective proteins. And that this overexpression or the activation of DOT1L expression and K79 dimethylation comes together with a disruption of the nucleolar architecture, probably not because of DOT1L, but because we reduce NPM1 levels. But both of them basically impact the expression of major satellite repeats, which we have observed. Which is the main story, right? You see, it's not so much on DOT1L. It's more characterizing NPM1, but we did not find really good evidence what both protein could do together in a complex.
I see. Mm-hmm. So switching gears a little bit, another protein next to DOT1L that you are working on is FOXG1. FOXG1 prevents cell cycle exit by binding to the SMAD phoxoprotein complex. And you investigated the details of this process. What did you find there?
Mm-hmm. Yeah, FOXG1 is one of these transcription factors that are of absolute importance for the developing forebrain, right?
Is it a pioneer transcription factor, right?
It is a transcription factor, yeah.
A pioneer transcription factor?
Pioneer, I would doubt that it's a real pioneer and being able to first open up and then basically allowing chromatin modifications to be placed. I think it is a modulator of the chromatin.
Oh yeah, sorry, I was mixing it up with FOXA1.
FOXA1 is a clear pioneer, right? But for FOXG1, this has not been shown, and also our chromatin data do not really propagate this pioneer function. So it's a modulator of chromatin interpretation and accessibility. Yeah. So FOXG1 is really important. We used it as a query line for studying DOT1L, and in this context, because it's a lock-in, we also thought, okay, it might be a good idea to study FOXG1 itself. And this is why this is a spin-off and being now an own line of research. FOXG1 has also many functions at the chromatin level, but also aside from the chromatin level. And we also studied this impact on post-transcriptional regulation, for example, by association with the microRNA processing machineries. And at the chromatin level, we were reporting earlier this year a large study of where we used also ChIP sequencing for FOXG1, also attack sequencing to understand what and how FOXG1 can influence gene expression at the chromatin level. FOXG1 is also mainly reported as a transcriptional repressor. As usual, we find that it's also a transcriptional activator, contradicting these earlier findings. But I think this is now also accepted in the field that depending on the context, FOXG1 can activate or repress. And we found a strong correlation to enhancer regions again, and a reduction of FOXG1 impacts chromatin accessibility and NK27 acetylation at these sites. And there we also found that FOXG1 is not only acting on its own, but that it has important interacting transcription factors such as NeuroD1. And there we basically found, which I find very attractive also as way to regulate transcription, that there is not a clear hierarchy between these two transcription factors, but that they influence each other. Sometimes it's NeuroD1 binding and recruiting FOXG1. Sometimes it's the other way around. And in some occasions, they also act totally independent from each other. So I think what drives us now with future questions is really how these site-directed recruitments and how these site-specific dynamics of transcription factors, of chromatin modifiers can be explained, right? Because we know that they are regulated and that they find their place, but we don't understand how that's going. And this is basically where the signaling comes into play. You have mentioned correctly that FOXG1 acts together with a SMART pathway. That means it's under control of the transforming growth factor beta family, TGF-beta. But this is not the only one. It's also controlled by FGF signaling, that means fibroblast growth factor signaling. And it will be really interesting and also one of our next attempts to understand how signals basically regulate these chromatin-specific events that are occurring for transcription factors, but also for chromatin modifiers.
So you touched many of the questions I wrote down. So maybe I can just pick some. So you mentioned the story that you published earlier this year, so 2023. And there you used, as you said, you used many methods together in a multiomics approach. So why did you choose this multiomics approach? What did it enable you to do? What was the real advantage here?
Yeah, that was a factor that delayed our publication quite substantially, because we did this FOXG1 chip, and I was really surprised to see it in enhancer regions. And then we were working in primary hippocampal neurons, and data sets are sparse and not many omic data sets published on this particular cell types. So we were forced to really show that these are enhancers, which then basically drove us into performing the K27 acetylation chip sequencing. But because we did not only want to do K27 acetyl, we also included H3K4 trimethyl and I think also 27 trimethyl within this data set. So that was the second multi-omics part. And then because we were then proving that we are working with enhancers, we said, okay, if FOXG1 impacts K27 acetylation at enhancers, it will probably lead to differences in chromatin accessibility. The way to address that is then to do attack sequencing, which was then the next step that we did. And in the course of this experimentation, we wanted to explain why K27 acetylation is changing, because FOXG1 is not a histoacyltransferase or a deacetylase. So it basically has to interact with something. So we did a proteomics analysis. We saw that HDACs associate with FOXG1. And then basically the question is, of course, if you work on a neurodevelopmental disease, of whether you can use this information to drive therapies. And we thought, okay, there are so many HDAC inhibitors, and if FOXG1 works together with HDACs, so maybe we can exploit this information for doing therapies. And there we then did an inhibition experiment of HDACs. And this, of course, meant that we had to do another data set and look for transcriptional alterations, which is basically, opening one box leads to many more boxes that you have to open to understand the biology behind.
At one point, you need to say, "Well, now it's enough, right? Now it's enough. We have done enough experiments. Let's write the paper."
Yeah, but there was one question after the other, and of course now we have a plethora of questions that we would like to ask using multi-omics approaches, which I find really, really fascinating to work with, because we always have a genome-wide view, and there are so many observations that you can draw, that you can observe in the single data sets. But if you then combine, you have other views on the regulative mechanisms, and to untangle these and to understand the individual molecular machinery, I find absolutely fascinating and driving our research.
Mm-hmm. So you already kind of answered my next question, but imagine you are due to submitting a grant proposal tomorrow. What would you have written into that, or maybe would have written into that if it was not coming from your lab now? So maybe a more imaginative grant proposal.
It's not that we don't have many ideas in our head. So obviously for FOXG1, we would like to understand or to come a little bit closer towards understanding the human disease, because human development is a little bit different from what we know from the mouse, of course. And we would like to get funding for driving our research on human FOXG1 and using brain organoids, and now putting basically everything that we have learned in methods, but also in the biological system from the mouse towards the human system. So that is what we are doing there. And in regard to DOT1L research, I would really, really love to understand whether K79 methylation has a meaning or not. But for this, this question is really difficult to untangle, and I don't think that I could write the grant just now. I'm always thinking that larger labs equipped with more technologies, they should tackle these difficult questions. But since nothing is basically being published, I think that other labs and other PIs or other researchers are as clueless as I am understanding this particular mark at this very moment.
So for the last almost 40 minutes now, we have taken a journey through your scientific career. Did we miss something important, or would you like to add something?
No, I think this is mainly the content, right, of my research. Of course, there's many things that we did not talk about. All the failed attempts, for example, that come across or come alongside our scientific careers, all these hypotheses that you find absolutely brilliant at the moment, that you think about them, but which you can never prove. This is, of course, also something that is part of any scientific career. Like probably people do not touch on these points too much, but I think we should be honest and also encouraging the younger and early research careers for not giving up, but to follow the path and to find the right boxes to unpack. And it's clear that sometimes a box is empty. Nevertheless, science is real fun, and you can ask so many questions. So, being curious is, I think, a main driving force.
So, thank you, Tanja, for your time and for being on the show.
Yeah, thanks very much for your time and having me here. And enjoy Christmas, everyone who might listen.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.